Hello, everybody. Thanks for joining the first Cancer IO Sebinar ex vivo models in IO research. We're super happy to host you today from literally all over the world, from Boston to Helsinki and pretty much everything in between based on what I've seen. And I'm your host and the chair of this session, Heidi Haikala from Dana-Farber Cancer Institute and Harvard Medical School, and I'm also part of the Cancer IO program. And just briefly, Cancer IO is a Business Finland network for IO research and for promoting the uptake of new IO drugs. And it's coordinated by the University of Helsinki. And if you're wondering what a Sebinar means, or if it's a typo or something, I've been asked about it a lot. It's actually due to the COVID situation, we're having everything truly virtual, but then when the world gets a bit more normal, then we're planning to have like a hybrid type of meeting. So seminar plus webinar. So we came up with the funny name Sebinar. And the Sebinar will be recorded and we will publish it in YouTube and our web pages and the LinkedIn event, if that's where you came from. And just a couple of practical things before we get started. We will have first 2 excellent short talks, followed by a keynote from Dr. David Barbie. And after the 3 talks, we will have a brief scientific Q&A for all the 3 speakers. So if you have any questions, just hold on to them and we will address them after the 3 talks. And then finally, in the end, we will have very interesting panel discussion that I'm looking forward as well. So we will talk about the applicability of these models that we describe here, possibly in the clinical decision making. And Mirjami Tranmin, whose name you might see somewhere here on the screen, she's from Cancer IO and she'll be moderating this QA chat and bring to me some interesting questions from there. And just remember to use the actual QA function. That's the way how you can ask the questions. If you use the chat, that is only to inform us organizers about something, so don't use that one. And if you want to reach the Cancer IO office team, you can do that throughout this webinar from the chat function or by emailing cancerio-office@helsinki.fi. And remember to use the official hashtags. If you use social media, you can see them on the screen right now. For the University of Helsinki students, you can get credits from these sessions by signing up in WebAudi and sending us a half-a-page short summary of what you have learned. Hopefully we all learned something here. And I think that's all from my side for now. So we will move on to our first speaker, who's Dr. Jeroen Pouwels, who is an adjunct professor and research coordinator in Cancer IO and in the University of Helsinki. And he's going to be giving us a brief introduction of what are these ex vivo models that we're talking about all the time. And yeah, the floor is yours, Jeroen.
So Miriam, do you stop sharing? Yes, thank you. I will share my screen. So good afternoon, everyone, or good morning, or good night, or wherever you are. Also, on my behalf, welcome to the first Sebinar from Cancer IO. My name is Jeroen Pouwels, and I'm, as Heidi mentioned, I'm the research coordinator of the Cancer IO. And today I will be introducing different preclinical cancer models to you. Obviously, in 10 minutes, I cannot go into depth for any of these, but I hope that I will be able to introduce all of them or most of them in very brief time, and then that will lay the foundation for the discussion later, and also maybe might make it easier to follow the other 2 talks. But before I go into the topic, I would like to first briefly introduce Cancer IO. Heidi already said a few words. It's a Business Finland funded big consortium. It has 2 universities, many hospitals in Finland, some small to mid-sized companies from Finland, many big pharma companies and some patient organizations. And we mainly focus on 3 themes: IO society, IO research, and IO healthcare. From the University of Helsinki, we have the Klefström lab where I'm working and the Mustjoki lab, and then Maja Holmen is with us from the University of Turku. That's all I want to say due to time restrictions, but please visit cancerio.org for more information. And also contact us for any information, or if you would like to collaborate or something, you can always reach us at cancerio-office@helsinki.fi. So the reason why these preclinical models are so important is, as you're probably all aware, is that most of the drugs that come out of preclinical development phase, they fail in clinical trials. This is especially bad in the oncology field, where actually 95% of the drugs fails. This leads to a tremendously high costs for drug development. So one drug costs over $2 billion to develop. And this again leads to increased drug prices for the consumer. I think that Johana will talk about this more a little bit in the next talk. And we feel that one of the reasons, not the only reason, but one of the reasons is that the preclinical testing phase does not adequately predict drug efficacy for the patients. And as you probably are aware of, there's many different methods that are being used to test drugs with different levels of complexity. And I will introduce several of these, the ones shown here today. And I will start with cell lines, which are the easiest ones. Most of you will probably have worked with 2D cell lines, which is the standard cell lines. They're easy, they're cheap, and they're fast, and they have a lot of other advantages, but we do know that they do not capture 3D biology. They do not represent the heterogeneous tumor environment. And most importantly, we know that drug responses in 2D do not, or very seldomly, reflect the response in patient tumors or tumors in patients. Some of these disadvantages have been circumvented by growing the cell lines in 3D. However, this is still not ideal, and they still do not represent the heterogeneous environment or capture the patient heterogeneity. One important recent discovery and development is the organoid cultures. When I talk about organoids in this case, I'm really talking about the system developed and pioneered by Hans Clevers, but at the moment already used all over the world by hundreds, if not thousands of labs. So here you isolate tissue from a patient or a healthy individual, you make single cells out of those, and then from the stem cells, organoids can grow out, which are like organ-like structures. For example, there's mini guts that can be made out of those, and they represent a 3D structure of the original tumor or tissue, and they've also been shown to really quite well predict drug responses. One big advantage of the organoids is that you can propagate them and you can create biobanks. And the paper you see on the left side, this Cell paper, was the first, to my knowledge, the first cancer paper on these organoids, which was in colorectal cancer. This is still the poster child for organoids, but many other tumors now have organoid biobanks and protocols. Also from metastasis, you can develop organoids, and you can also do this from circulating tumor cells, and many other organoid types are present. However, and these are really great systems. However, they still do not represent the heterogeneous tumor microenvironment. And why do I keep banging out about these tumor microenvironment? Well, the reason is, as you're probably all aware, a cancer is not just cancer cells, but it's comprised of a lot of different cell types. For example, cancer-associated fibroblasts, immune cells, and many others. And they're not just bystanders, these other cell types, but they very actively influence how the tumor develops, how fast they are involved in drug resistance, and many other things. One important aspect to highlight here are the immune cells. The reason I want to highlight these is that because most of the drugs currently in development and in pipelines for cancer are immune drugs or immunotherapies, which means that they do not kill the cancer cells directly, but they try to wake up the immune system in the patient and have that attack the cancer. And the big advantage of these immune therapies is that they can cure a patient rather than delay the inevitable growth of the tumor. This happens only in 10, 20, or 30% of the cases, but still it's a huge step forward. And obviously to test these immunotherapies, you need to have an immune component in your model system. For organoids, because they derive from stem cells, cancer stem cells, they do not have an immune component, but there's a lot of developments ongoing here to implement the immune system here. One of this is where the organized or grown together with lymphocytes from the tissue or from the blood, but we still feel that this is not the real situation. Another way to obviously implement a immune system or include an immune system is to use whole organisms, and obviously mice are used a lot. And most of the drugs that are currently in clinical use have benefited tremendously from mouse models. There's immune deficient mouse models where you use human cell lines and inject them into the mouse, or you use these patient-derived xenografts, but as mentioned, these are immunodeficient mice, so you cannot really test immunotherapies here. The other option is to use mouse cell lines or then genetically engineered mouse models to develop tumors in the mice. And since these are mouse tumors in mouse, they can be done in fully immune competent mice, and you could use these to test immune drugs or immunotherapies. However, a running joke among health researchers is that everything has been cured in mice. This is not to say that, as I mentioned, mice have been extremely important in drug development up till now, but there are clear disadvantages. They have a non-human context. Most of the drugs developed are being developed against human proteins, and they are not always recognizing the mouse proteins. Research mice are inbred, so they don't capture any genetic variation. Some of them are immunodeficient. They're high cost and slow. And most importantly, we know from experience that a lot of the drugs that work really effectively in mice, they simply do not work in humans. Some of them do, but a lot of them don't. And so the last model I want to introduce is something that we have developed in the lab, but many other labs have done this as well. In fact, David Barbie's talk soon will focus on these, on something similar as well. So we call them the patient-derived explant cultures. So these are explants. So we get tumors from the hospital several times a week, and we store part of them for biomarker analysis, but most of it is incubated with collagenase overnight. And what that results in is these small intact tumor fragments. So these are tumor cells and immune cells and calves and all of that. Obviously, there's no circulation here, but other than that, it's an authentic piece of tumor. We can culture these in the slides that you see, and generally we can get about 16 wells out of one tumor. And with several explants per well, we can then treat them with drugs, and we can have many, many different endpoints here. So single cell sequencing, immunofluorescence, and a lot of other things. And Heidi Heikkila, the host of today, she published a paper in our lab in 2019 in Nature Communication, so check it out if you're interested. But there she could show that a certain drug combination effectively kills MYC-high breast cancer, but not MYC-low breast cancer, or what we call healthy tissue, which is then either the adjacent tissue next to the tumor, or then tissue that we obtain from adduction mammoplasties. Another project in the lab by Rita Turpin, who is listening to this talk as well, is that do these PDACs now retain the immune contexture? And I will only show very few of her data here, but she could basically show that these immune cells are preserved after 1 week in culture. The basal activity is also preserved. When we add immunocult, for example, to these explants, we see that certain cytokines are being produced, or produce more or produce less, indicative of changes in the immune activity. And importantly, when we activate these, or when we add immunocult, we see killing of the cancer cells, presumably by the T cells who are activated by the immunocult. So we are currently using these explants with other more investigative drugs, and to see, can we model their activity, and can we learn new things from these drugs? So before I go to the final slide here on the acknowledgement, so this is all done in Joa Klefström's lab in Helsinki. Paulina and Heidi are thanked because they are the godmothers of the PDAC system, or the explant cultures, and Rita, you see highlighted here, who did all these experiments. The last slide I want to show is maybe a bit controversial, where I try to compare the different immune systems by giving simple pluses or minuses and judging different aspects of them. And here I don't want to say that one system is better than the other, but what I want to highlight here is that you probably need a combination of these model systems to truly do correct or good predictive drug development. For example, if you look at the explants that I talked about last, they have very many advantages. They are low cost, they are very fast. We have the results in a week. The microenvironment is mimicked or is preserved, and we have full patient variation, but our disadvantage is that the throughput is quite low. We can only do 16 wells. Organoids, they can go very high throughput, but then they lose some of the microenvironment aspects, and all the other models, they have certain weaknesses and strengths. And so my basically take-home message here is that, you know, we need to combine different model systems to do proper drug development. And with that, I would like to end my talk and give the microphone back to Heidi.
Thank you, Jeroen. That was actually an excellent bridge for the upcoming panel discussion. So we will be discussing about the models much more in the end of this show. And also, if you have any questions, hold on to them, or you can post them already to the Q&A function. So we will be addressing them after the 3 talks. So moving on, next up is Johana Kuncova-Kallio, I hope I said that right, from UMP Biomedicals. And she's going to tell us about some new interesting solutions for 3D tissue culture and for the preservation and shipping of tissues.
Hello, my name is Johana Kuncova-Kallio, and I'm the director of UMP Biomedicals. I think it's important to start from the challenges that the pharma industry is facing today. First of all, it's not very cheap to develop a new drug. On average, it takes 1 to 10 billion euros per single drug to enter the market. It also takes a lot of time. On average, 10 to 15 years of development. On the other hand, there is the constraints of the patent validity, which is 20 years, and if you are lucky and are able to extend it, you can get to 25. So that gives you 5 to 10 years of sales. So pharma is actually facing a lot of pressure in terms of how to make the drugs cheaper and faster, and that means how to avoid late failures. Actually, the best place to start is on the beginning. So in the phase when you are screening from 10,000 compounds down to maybe 5 that are entering the clinical phase, that's the good place to fail, not later. So failing early when it is cheap is one of the reasons why there is the shift towards 3D cell models and also the ways how they can be reproducibly produced and transported. So how to get there to the reproducibility? First of all, it's about the composition, then also maintaining the cell functionality, getting some automation into the process, also avoiding the freeze cycle. And then when we are looking a step further, it's about the translation of the results to clinical use. And that is something I will introduce briefly in the next slides. So first, composition. In our case, Grodex is made out of nanocellulose, which is made of birch and water. So there is no additional growth factors, no surprises. It's day to day, month to month, year to year, always same composition, which allows you also then to fine-tune the matrix as it is empty. You can add the ones that are actually needed for your cells. So when working on 3D cell models, it is also important to consider how long the cells are still okay to use. And for that, we have actually figured out that nanocellulose has been able to sustain the functionality of cells for a prolonged period of times, and also it does not alter the functionality of the cells. So that comes important when we are talking about patient-derived cells or stem cells or other more sensitive cells. And I have a few examples here. So this example of 3D cell culture from cancer patient-derived cells, which has been actually performed here in Finland by Vilja Pietiläinen. We have also example of patient-derived bronchial epithelial cells and dendritic cells, which is from Doris from Innsbruck. We have also smooth muscle cells and endothelial cells, a nice comparison with Matrigel and Grodex, which is from Teresa Mimler in Vienna. We have also an excellent work from Jan Bruder from Max Planck Institute, which is concerning neural organoids for high throughput applications. And again, sustained growth for 22 days in this case. Osteosarcoma cell line spheroids in Grodex T in this time from University of Helsinki, David Rice. Breast cancer cell spheroid, the growth of the spheroid, but also secretion of extracellular vesicles. In Grodex, this is work from Kirsi Rilla from University of Eastern Finland. 3D culture of leukemia cells from Jarkko Koivunen, University of Oulu. Ex vivo modeling of drug efficacy in rare metastatic urachal carcinoma from Juha Rantala in Miss Week Biology. Mini liver spheroids for high throughput screening from Brinton Seashore Ludlow in Karolinska Institute. So over the years, we have developed many protocols for 3D cell culture. This was just a little overview. And we have pretty much worked on every organ there is and many types of different cells. Third, for the reproducibility, it is also important to automate a bit. So our gel is compatible both with hand pipetting, but also with robotic handling of the gel, which is important when we are really thinking of doing any screens for big pharma for new drug. It is also important to remember that nowadays we are talking more and more about not only single cell type, but many different cell types. And here we have also opportunity to do this in a little bit more defined manner. So the top row is an example with 3D printed tissue. In this case, it is actually a liver. And on the lower side, we can see dispensed layer by layer different cell types for skin model. But our vision for drug screening is actually going beyond just the cell model. We are thinking of it as a workflow starting from biobank or hospital where you have a patient and then the cells derived from the patient, which are then dispensed or 3D printed into a well plate or organ on a chip. And then comes the critical point, which is transportation, because we believe that when you freeze the cells, half of them are not waking up on the other side, and that's really a lot of waste of valuable time and effort. On the other hand, pharma is a bit reluctant to develop these very specific models in-house. So I think this transportation plays a key role in enabling commercialization of these 3D models, which are developed in academia and their translation to actual big pharma. So I will come back to the transportation, but when the model is in the place of the instrument, whether it's high throughput or high content screening, that model can be directly screened. After that, we have Grodase enzyme, which actually allows us to release the cells and just get rid of the non-cellulose. And after that, you can continue to sequencing or another method that you use. So for this transportation, we have a nice example with neural organoids, which are quite sensitive, which have been grown for 22 days in Grodex. And after that, they have been shipped at 37 degrees in a cell box, which is an incubator for transportation of the cells around the world, really. So it's actually allowing the model still to grow on the way instead of freezing it and kind of putting everything at stop. In this case, you can then continue the workflow as if nothing happened, just in different laboratory, in different country. Next is from lab to fab. So we have been working on this gel from the research level, which didn't have really much of standardization. We have developed it. We have actually a product which is a medical device on the market and is following the ISO 13485 standard. And now we are working on the next level, which will be a compatibility for use inside human.
Okay, for everybody, there's a small glitch in the pre-recorded video, so don't be alarmed. Johanna will return in a little bit.
Regenerative medicine is really that the nanocellulose would not be just a component that is used, for instance, during testing or just in one part of the workflow, but actually it would be the carrier for the cells from the biopsy in the patient through the refinement, bioprocessing, through testing, and then application back as a therapy for the patient. And we have different methods or different forms in which we can apply it, whether it is injected as a gel or it is applied on a wound dressing. It can be 3D bioprinted. It can be also released. The cells can be released from the gel, or then we have an extra card for freeze drying solutions in the gel. And this is one such example where there is transplantation of HESC-derived otic neural cells to the inner ear, which have been then injected with a fine needle. UPM Biomedicals is the forerunner in producing high-quality nanofibril cellulose for medical and life science applications. We wouldn't be able to do this alone. We collaborate extensively with universities and research centers, and also with the key industrial partners. We are part of UPM, which is a more than 100 years old company. We have more than 300 patents in the portfolio covering the nanocellulose and its use, and we have today 4 nanocellulose product families, including GrowDex for 3D cell culture, where we have 3 different products, Growing for 3D cell printing, also 3 different products there, and FibDex, which is a CE-marked advanced wound dressing used in hospitals. We have more than 60 projects with academic and industrial partners. If you want to find out more, you can visit our website upmbiomedicals.com or directly growdex.com. And of course, I'm staying here for further questions, and also so do my scientists.
Okay, thank you, Johanna, for the excellent talk. It seems like a very innovative approach, and I guess we can address in the panel as well if this is a vegan solution also for a lab 3D culture. But moving on, so the QA will follow after the talk. So moving on in the program, so next up we get to our keynote talk from Dr. David Barbie. And Dr. Barbie is an assistant professor of medicine in Harvard Medical School and thoracic oncologist in the Dana-Farber Cancer Institute, and also an associate director in the Belfer Center. He obtained an AB degree from Harvard College and an MD from Harvard Medical School in 2002. And Dr. Barbie was an HHMI medical fellow working in Dr. Ed Harlow's labs in Mass Gen Hospital. And then he did a medical oncology fellowship in the Dana-Farber Partners Cancer Care Program, which he completed in 2008. And this was followed by a postdoctoral fellowship in Dr. William Hahn's, so now a Nobelist Hahn's lab in the Broad Institute, after which he received the tenure track independent investigator position in Dana-Farber. So the floor is yours, Dave. Dave will be telling us today about ex vivo systems incorporating the tumor microenvironment.
Thank you so much. And you can see my slides just fine?
Yes.
Great. Yeah, these are my disclosures. And so I'm going to divide up this talk into several sections. And I think the previous talks were excellent and really helped set the stage for what I'm going to talk about. And I think one of the key points I like to make with immunotherapy is that it's very different from targeted therapies that I'll compare in a moment. And that the issue is that the immune system is dynamic, and that's what needs to be captured. I'm also going to do a review, which was as was done the talk just prior to the last one, about other models compared to ours which include patient-derived xenografts and organoids. But what I'm going to focus on for the majority of the talk is a system that we developed using organotypic tumor spheroids, and a recent publication where we've been refining the readouts of this system. So as I mentioned, it's in lung cancer, which is one of my clinical focuses, it's pretty straightforward to perform sequencing to look for mutations such as an EGFR activating mutation, which guides the use of an EGFR tyrosine kinase inhibitor. But when one thinks about checkpoint blockade, a similar biomarker, PD-L1, is not a static measurement. It's actually a dynamic molecule that can be induced on the cell surface. Not only that, the interaction of PD-L1 with its target, PD-1 on T cells, is also dynamic. And that's what's influenced when one treats with a checkpoint inhibitor that basically disrupts this interaction. Over time, T cells then migrate into the tumor, attack. And the point is that not only is this a dynamic event, but that there are multiple other cells in the tumor microenvironment that are impacted by this. There's also local lymph node trafficking and antigen presentation that occurs over time, as well as dynamic upregulation of other immune checkpoints. So the point is that what's currently being done, which is often a static biomarker measurement, does not reflect the fact that these dynamics are not captured by a single point in time. And so there's a number of papers that have been coming out over the last several years, giving us more and more granular measurements of the tumor microenvironment. These are several of them, one of which I'm an author on, but using high resolution single cell RNA sequencing. And for example, the top papers in melanoma, the bottom in breast cancer. And then there's other papers looking at T cell subpopulations and whether these can predict subsequent response to checkpoint blockade. But while these and other studies provide improved resolution in so-called tumor and immune atlases, most of these studies lack a time dimension. And in fact, there were 2 papers that just came out in Cancer Cell and Kidney Cancer that do start to look over time with what happens with single cell as patients are receiving checkpoint blockade. This was done actually quite early on in one of the very first papers showing efficacy of PD-L1 blockade. This is atezolizumab, the drug developed by Roche and Genentech. And what I'm showing here is a patient where they received a biopsy pre-treatment with checkpoint blockade and then on treatment 4 weeks later. And you can see that very clearly over time, you see CD8 T cells now coming into the tumor. And moreover, if you look at gene expression markers over time, you see that over time, you see induction of multiple cytokines and chemokines and activation markers of CD8 T cells. And this is in a responding patient. And so they then looked, can you find over time markers of patients that do or don't respond? This dynamic measurement, actually one of the most robust markers was actually PD-L1 itself. And this is not the fixed baseline measurement of PD-L1, but rather over time does PD-L1 increase. And you can see that in the patients who had the best response, that on the repeat biopsy, they saw an increase in PD-L1. And that has to do with the fact that I mentioned it's a dynamic marker, and actually interferon gamma can upregulate it. So this is likely just a reflection of the fact that over time, when you have an interferon response, you get this feedback upregulation. Whereas patients that did not respond did not have a significant increase in PD-L1 over time. But as you can see, you're not going to capture this in the baseline biopsy. There have been other studies, again, preceding the Cancer Cell papers I just mentioned, that did pre and post gene expression profiling, bulk profiling, pre and post nivolumab therapy, and looking for these similar dynamic markers. And indeed, as one might expect, consistent with this early study, that in patients that respond, you see activation of T cell markers, activation markers like granzyme A and B, as well as some other compensatory immune checkpoints. However, the issue with repeat biopsies is that it's invasive. And the other point is it lacks predictive power. You've actually had to treat the patient before you've figured out whether they're a responder or not. So that leads us to the focus of this whole topic, which are, can we start to develop ex vivo models that can be predictive of whether a patient will respond or not to an immunotherapy? And so when one thinks about and again, this was a really great overview previously. I'm going to kind of dive in a little bit in more depth because I have a little more time. Patient-derived xenografts and organoid models. What one basically does is they take a freshly explanted tumor from the patient, and maybe in the panel we can talk about this, that really a key bottleneck here is being able to acquire freshly explanted tumors, that most of the time it's immediately fixed or frozen. But what's required is a media actually in the operating room or in the biopsy suite. And simply a piece of that tissue is then preserved live in media. And then when it's brought to the lab, it can either be enzymatically dissociated completely to single cell suspension. And this is what's done in the studies I showed of these immune atlases, where you can perform flow cytometry at baseline or single cell RNA sequencing. But what's done in PDX and organoid models is that these suspensions are then implanted either under the flank of an immunocompromised mouse or grown in special media to favor cultures of organoids. And as was alluded to, what happens in both cases is that you get tumor-specific expansion, and it's on the order of weeks to months. And as I'll show in the next slide, the particular issue with PDX models is that you need to deal with mouse-specific immunity. And that is not to say that these models have major limitations, as was also alluded to. Their strengths and weaknesses is every model. And in the humanized, in the PDX models, there are a number of efforts that are ongoing to allow so-called humanization and compatibility, because one might imagine the major issue if one is trying to add human T cells into a mouse is graft versus host disease. However, in the time scale that these studies are done, it's often not a major issue. And there are also solutions that have been developed, genetically modified strains that resist GVHD. Another major issue is HLA restriction and compatibility. There are a number of models that are being developed to counteract this. But I think one kind of underappreciated issue is that human cytokines are different from mouse cytokines. And so that's a very difficult thing to control, but there are also mouse strains that are being developed to sort of humanize the cytokines. But just as an example that this is possible, there are several publications in melanoma, for example, where one grows the PDX as is typically done, and in parallel harvests the TILs, the tumor infiltrating lymphocytes, cultures them with tumor cells to expand those T cells that are capable of recognizing tumor cells along with IL-2. And then after a rapid expansion, infuses them into these mice in an adoptive transfer model. And what they were able to show is that particularly with continued IL-2, that when one adds pembrolizumab, that you can actually see a response against the human melanoma tumor that's specific to treatment with pembrolizumab, checkpoint blockade. Another major approach to humanization is developing mice in which you actually reconstitute with human bone marrow. So you isolate human CD34 positive stem cells and reconstitute these severely immunocompromised mice with a human immune system. And here again, it's been shown that treatment with nivolumab can lead to responses in the humanized context. And as well, that in the presence of nivolumab, you actually see these humanized tumor TILs basically infiltrating into the tumor. Following up on the Hans Clever's model introduction, work out of his lab in an adjacent lab also showed that one can grow a mismatch repair deficient colorectal cancers that are known to respond to checkpoint blockade, and do this similar procedure where you grow the organoid, but also take, in this case, peripheral blood T cells and expand them, and then basically screen these T cells that you grow for activation and tumor cell killing. And what was found is that in a number of the organoids and in the patient-matched specimens, you can actually successfully identify subpopulations of CD8 T cells that are able to actually recognize the tumor. And that this is definitely specific because when they block with an MHC1 blocking molecule, you can rescue this. And then finally, work from Calvin Kuo out at Stanford has similarly worked on growing organoids and basically looking at T cell expansion with organoids and then stimulating specific T cells with PD-1 blockade and finding that you can actually do the same. You can get basically checkpoint responsive T cells to recognize these organoids. But as was mentioned, there are certain strengths and weaknesses of each of these models. So one of the key strengths of the PDX, humanized PDX and organoids-based technologies is that the amount of tissue is rarely limiting, that you can expand over time and basically conduct a large number of experiments, and that it can be useful if one is very focused on actually the reactive T cells, which are the ultimate effectors of most of our immunotherapies, that one can match antigens with T cells. However, that one loses, as was also alluded to, the many different heterogeneous cells that are present in the original tumor immune microenvironment that are likely highly relevant. And moreover, when you're expanding the tumor, you're also potentially expanding out subpopulations that may not reflect the original tumor heterogeneity. And finally, I think what's most important is that in a refractory setting, where you need a rapid timescale for decision-making, that it takes months to expand these tumors, perform experiments, that one might not have that time. However, as these technologies move into early stage cancers, i.e. high risk cancers that have been treated either with surgery or radiation and chemotherapy and potentially cured but have a high risk of relapse, there you do have time to conduct these kind of technologies. So, but what I'm going to focus on for the rest of the talk is work that came out of our lab now several years ago around patient-derived organotypic tumor spheroids. And this, as long as what was derived from, explained earlier with the explant model, I would say falls into this category of functional precision medicine. And this is a review that was a very nice review by Tony Latai, who's also at Dana-Farber. He published this in Nature Medicine several years ago. He's actually founded a Society for Functional Precision Medicine. And basically what he likes to highlight is what I just did about at the beginning about targeted therapies, is that even with targeted therapies, EGFR and others, we can't be 100% sure that the patient will respond. So why not develop a functional readout like we do in microbiology, where you actually test susceptibility to an antibiotic? And this concept has been around for many, many years, but I think a major theme is that new technologies really begin to make this a reality. And so in his case, what he's really focused on is tumor cell killing. And basically they've developed a way to measure apoptosis through really what happens at the level of the mitochondria. They call it mitochondrial priming. And so where one, as I mentioned, for targeted therapies really focuses on these static omics analyses, genomics primarily, and identifies a mutation for which one can target, that here one can, especially if you don't have a target, or even if you do, if you can keep the cells alive for a short period of time, treat, and then in this case, look at killing. And again, the advantage here is that you get a readout rapidly, basically from the biopsy to readout, in this case, within a few days. And so the way that we stumbled into this area ourselves is that we had been very kind of frustrated with standard two-dimensional culture. I've had a longstanding interest in innate immunity and how cytokines might affect biology of cancer cells. And we realized that you need to develop 3D spheroid models to start to look at the effects of cytokines on tumor growth. And we had this very simple idea that we were already using filters, that basically when you use cell culture to capture spheroids, basically you grow cell lines on low attachment plates, and you basically use filters of typically what my postdoc was using were 40 micron or 100 micron. And this was simply a way that you can capture 40 to 100 micron cell line spheroids for growth in a microfluidic system. But we said, well, what if you actually apply this to tumors? And what I'm going to show you are both, I'm going to focus a lot on the murine-derived models because we can validate the efficacy of the system in mouse, but also from patients. Basically, the same procedure I mentioned, one takes a fresh tumor specimen and tries to immediately put it in media so that it's you get very little tissue ischemia and necrosis. Bring it to the lab, but instead of complete dissociation to single cells, we basically mince the tissue and partially digest it using a variety of different enzymes, such that we don't break it all the way to single cells. And then we simply apply the same procedure using these filters. And what we realized is that when you capture these 40 to 100 micron spheroids and you now run flow cytometry on them, that they actually have all the components that were in the original tumor, not only tumor cells and not only immune cells, CD45 positive cells, T cells, B cells, myeloid cells, but also stromal cells and even a small percentage of endothelial cells. And so what we do is we take this fraction and then we basically load it, and I'll show you a picture on the next slide, into a microfluidic culture system that was developed by Roger Kam at MIT, where this is a central 3D, this is a top-down view of a central 3D collagen channel, and these are the spheroids that we embed. And so this is using one of our mouse tumors, MC38, which is in a syngeneic, it's a colorectal syngeneic model that's known to respond to checkpoint blockade. And so you capture these spheroids, and this is again a top-down view. You reestablish them in collagen, and you can see that basically over time, these spheroids expand. And this is, like the explant model that was described earlier, a short-term model that we end the experiment after 1 week. A, because we can show that we retain immune cells up until this time point, and B, as you can see, what starts to happen as these tumor spheroids start to secrete factors that break down the collagen. And so often if we try to let it go too long, you'll get collagen collapse, and we need to end the experiment at that point due to that reason. But over this period of time, what one can do is then treat with various perturbations, such as checkpoint blockade. And in addition, what the system allows you to do is, because there's media ports on both sides, you can capture the media. And what I'm showing here is just the baseline media. And what you see is that, consistent with the number of immune populations that these cell spheroids contain, is you can actually run Luminex cytokine profiling and see that these spheroids produce high concentrations of multiple different cytokines and chemokines over this time period. And so we then wondered, could we treat with PD-1 blockade in this model and see both a live dead response as well as a cytokine response? And I'm going to focus on the live dead response because we think that's most interesting in terms of predictive value. What we used here were the mouse models, because if you take it from a patient, it's harder to control response. You have to do a much larger study to really prove that ex vivo response correlates with in vivo response. But in the mouse model, we already know which of these mouse models respond to checkpoint blockade. And so we used, in this case, what I'm showing you is the MC38 model, which is known to respond. We treat with a control antibody or the anti-PD-1. We culture over time, and then we use dyes, a green dye, such as calcium, and a red dye for dead cells, such as propydium iodide. And you can see that, as has been published many times, that in vivo, when one treats the MC38 model with PD-1 blockade, we see killing, but not cures, which I'll come back to later. And in our ex vivo model, when we harvest the spheroids at this time point, after 2 weeks after the tumors have formed, and then treat ex vivo with a control or a dose-dependent concentrations of a mouse PD-1 antibody, by day 3, we see very little killing, but by day 6, you see a dose-dependent, time-dependent killing with checkpoint blockade. And we've done a number of experiments to show this is actually CD8 T cell specific. If you neutralize with a CD8 antibody, this effect is blocked. But also, very simply, if you simply take the cell line without immune cells, grow them as spheroids, and run the same assay, you don't see any effect because, again, there's no immune cells. So this just shows this is not a non-specific effect. We've also now extended this in the paper that we published to multiple mouse models as well as human models. And you can see that models such as the B16 melanoma model that's highly resistant to PD-1 blockade has immune cells, which we can show, but doesn't respond to checkpoint blockade. And we tested a variety of other resistant models, but what I want to focus on, one of the models we like to use is a CT26, which is also a colorectal model that is partially sensitive or partially resistant to checkpoint blockade, because then one can test combination therapies. And so my lab has had a very long-standing interest, as I mentioned, in cancer innate immunity. When I was a postdoc in Bill Hahn's lab, through RNAi screening, we found a role for the kinase TBK1 in promoting survival of KRAS-driven lung cancers. This is a crystal structure of the TBK1 kinase, one of the first papers in our lab in collaboration with Michael Eck. And this is a very important kinase. It's actually gained a lot more attention recently downstream of the cGAS-STING pathway. But it basically gets activated in a variety of different contexts to activate an interferon response, NF-kappa B response. It's also been implicated in autophagy. And as I mentioned, it can be co-opted by oncogenic KRAS. And so we had been working with Gilead Sciences on developing more potent and specific TBK1 inhibitors. And we realized that some of our early interests in this pathway in tumor cells, that what happens is this kinase and its homolog can make these pro-tumorigenic cytokines that are driven by NF-kappa B, that at the same time papers were starting to come out that in T cells, there is a particular function of this kinase. It turns out it's downstream of the STING pathway. And that if you hyperactivate this pathway in T cells, you actually kill them. And so Gilead, and also there were papers coming out that if you knock out TBK1, you can hyperactivate T cells, had basically found that with this compound, you can actually not only inhibit tumor cell cytokines, but actually activate T cells. So we realized this could be a very nice compound to test and potentially resensitizing to checkpoint blockade. And indeed, this was the case, that in this resistant model where we saw ex vivo with the PD-1 alone, very, very modest activity, red is the dead quantified, and with, here with the compound alone, very minimal killing, with PD-1 alone, very minimal killing, but with the combination, we saw cooperativity, and here you can see, this is just the images of the live dead, that we see this synergy. And the point is then when we went back and did the in vivo experiment, this predicted that in a mouse now, that you could predict this combinatorial response. So this basically starts to say that one could use spheroids, not as the be all and end all, but if one wanted to test a variety of combinations for which are most effective, and then go back to the mouse to validate which ones are the most effective, that this is a nice system. And again, whereas in human tumors, it can be somewhat limiting, in the mouse, one can generate a lot of material to do this. And so what we've obviously been focused on, though, are human samples. And this is just to show my interest is in lung cancer, that we can culture these same, this is from a non-small cell lung cancer patient, just showing that we run flow cytometry, but also in the device. EpCAM is the tumor spheroid marker, cell marker within the spheroids, but these spheroids contain live CD45 positive immune cells. And when you stain for CD8 in the devices, you also see that they contain CD8 T cells, and that one can occasionally see anti-PD-1 responses in these spheroid models. However, compared to the mouse system where one sees a rapid response, it's much more often that we see a cytokine response to checkpoint blockade in this 1 week timescale to PD-1 blockade. What we have become very interested in using these models is perturbing innate immunity. And so one can treat with interferon gamma and measure the response to interferon gamma. And it's interesting that actually a number of there's heterogeneity in this, that there's actually sensitivity to when you drive an interferon response. And actually one of the things we're mainly focused on now is the STING pathway, and that one can treat these spheroids with STING agonists and look at cytokine responses, which are very good in human tumors over the short term. But what I want to focus on in the remaining portion of my talk, and to make sure we have time for the panel discussion, is how we have been working on refining readouts. Because to date, what we had published on was live dead imaging and the cytokine profiling. And this is now a more sophisticated view of what we've been working on. This is in collaboration with the Belfer Center, which Heidi mentioned I'm a co-director of with Dr. Pasiene. And my colleague Cloud Powlitz, who is the head of the Belfer, has been very instrumental in helping to really help to industrialize this technology within the Dana-Farber. And so now we can do this in, again, patient-derived samples. We call them PDOTs, the murine-derived I mentioned, and these syngeneic models, MDOTs, and what Cloud is calling XDOTs for xenograft-derived, where you can actually test targeted therapies. One captures these spheroids, we load them into this device, as I mentioned, we grow them over time, treat with a control or the drug. What we do is we run flow cytometry, and what we found is that at baseline, if you compare these different fractions, the proportion of immune cells in our spheroid fraction matches that of a single cell fraction. So we can get away with preserving the spheroids, yet measure the immune composition as a proxy through these single cells. And then, as I mentioned, in the device, we can then verify that we see similar proportions using one device sustained for example, CD8 T cells. But we're working on developing continuous imaging methods. But what I really want to focus on here is the power of now performing RNA sequencing at the endpoint. And so this is a work that we just published in JCI with Kartik Sehgal, who's a very talented postdoctoral fellow in the lab. This was also highly collaborative with the Belfer Center as well as Novartis Oncology. And what I'm showing here is the MC38 model that I mentioned is a highly sensitive mouse model to PD-1 blockade, but not cured by PD-1 blockade. And I'm recapitulating some of the data that I already showed you. And we basically harvest these MC38 tumor spheroids, we grow them in the device, and we treat with anti-PD-1. And as you can see, these are sensitive to PD-1 after 6 days, as I mentioned. Here is the quantification, here is the individual spheroid pictures of the dead being read. But what we wanted to do is a couple of other things. We wanted to compare what happens when you treat with interferon gamma by itself. And in this case, this is not an interferon gamma, this tumor type is not killed by interferon gamma. So you can see there's very little effect in viability. And here I'm also showing you that when we add a CD8 neutralizing antibody, we can rescue the effect of PD-1 blockade. But so what we wanted to do is then run RNA sequencing. This is bulk RNA sequencing in all 4 of these conditions and compare what happens to the gene expression profiles relative to the control. And what was quite interesting is even though we didn't see a cell death response with interferon gamma, when we run this is a volcano plot looking at the log 2-fold change in genes that are either upregulated or downregulated by interferon gamma, and this is the significance, you can see that there's a number of highly significantly upregulated genes, and these are all known interferon gamma targets. In fact, this GBP2, 3, 4, 7 really map out interferon gamma transcriptome. And then you see one of the key chemokines induced by interferon gamma, CXCL10. And when you run gene set enrichment analysis, it's clear that we treated these cells with interferon gamma. It's the top response signature, and you also get overlap with interferon alpha response signatures. So this was nice, saying that basically we can successfully recapitulate gene expression profiles with the expected perturbation. And so we're very curious, though, what happens when you treat with checkpoint blockade? And what we noticed is that there was a unique pattern of genes that gets introduced. Not this interferon response that we might have expected after 6 days, but rather a very strong signal of EMT. And you can see here that SNAIL1 is upregulated, and also a number of genes that are known to be involved in promoting this stem-like phenotype, LIF, which is an IL-6 family member, as well as indicators of IL-6 activation. SOX3 is a negative regulator that gets activated upon IL-6 induction. And we, in fact, confirmed in the media that IL-6 is being induced, even though we didn't see it at the gene expression level. And so this was all recapitulated in our gene set enrichment analysis that we get this NF-kappa B signature that was actually captured by a TNF signaling via NF-kappa B, which I'll come back to, as well as EMT being the second induced pathway. But what we realized, however, is that we're profiling at day 6 when actually most of the tumor cells are dead. So while we were initially thinking this could be a signature of actual PD-1 blockade, what we also realized this could be is a signature of the cells that are surviving after PD-1 blockade. And this concept has been well described for EGFR, TKI therapy, and others of drug-tolerant persister cells. And we wondered, could these be actually so-called immunotherapy persister cells? And so to do this, we wanted to develop methods to actually liberate these remaining cells, because again, bulk RNA sequencing is really just digesting the entire population. And this is technically challenging because what one has to do is at the endpoint, instead of just adding the lysis buffer, which we do for bulk RNA sequencing, we have to liberate with collagenase to break the spheroids out of the collagen and then actually add trypsin to convert to single cells for 10x single cell sequencing. And I will say one of the current challenges with this methodology is even though we can visualize immune cells in the device at the end of treatment, after these treatments, we basically lost immune cells because it's so harsh. But in our case, this was a useful trick because what we retained were tumor cells. We knew these were MC38 tumor cells with some small populations, but a very minority of stromal cells. And what was nice is that when we did this with the control IgG versus PD-1, and these are the high dimensional reduction, so-called Tisney plots that one can generate and cluster, whether there are patterns. What was very gratifying is that the anti-PD-1 treated samples clustered very distinctly from the IgG treated clusters, suggesting that there were unique gene expression profiles of these cells that survived after PD-1 blockade. And to summarize a lot of data, you can see that there are 4 different clusters here within the anti-PD-1 residual cells. And when we looked at that EMT signature from the bulk population and projected it onto this space, what lit up was this cluster 2. That was what was most enriched for this EMT signature. And what also uniquely separated out this cluster was that it, as opposed to cluster 1, downregulated cell cycle genes. That cluster 1 had a number of E2F target genes and mitotic genes, whereas cluster 2 was starkly absent for these, suggesting that what this population was an EMT positive quiescent cell population, potentially that could represent these so-called immunotherapy persister cells that we coined. So to study this further, we use this EMT signature. We would call it an EMT-like signature because it's really a hybrid signature similar to what Bob Weinberg has talked about, because it's like a stem-like signature that doesn't have the full markers like Vimentin of an EMT state, which is why we call it an EMT-like state. And then we basically use this of our single cells to say, okay, which are the single cells that had the highest expression of this signature versus the lowest, and can we find marker genes of these EM-like cells? And what we found was that, of course, genes within this signature showed up. These were actually genes in the signature. But what was nice is that SNAIL1 was actually not part of the signature at all, yet it was clearly enriched in these in this cell state, as was this other gene, which is known as SCA1, also known as Ly6A, which turns out is a hematopoietic stem cell marker in mice. This to validate, now that we have these 2 markers, that this could actually happen in vivo, we developed a SNAIL1 in situ hybridization, and you can see that indeed in vivo in MC38 treated tumors with PD-1, you see that especially in the areas of undergoing cell death, you see accumulation of SNAIL1 positive cells. But what was most useful was the SCA1 marker, because we initially had some skepticism that were these potentially contaminating cells, these SCA1 positive cells, stromal or other. But we had evidence from literature from the lung cancer literature, actually. Carla Kim, who trained in Tyler Jack's lab, found that SCA1 can actually mark in the KRAS lung cancer model, lung cancer stem cells. And so no one had actually ever looked at this in the MC38 cell line itself, or in the CT26 cell line itself. We could use the SCA1 marker to see, are there actually subpopulations that pre-exist in these cancer cell lines? And indeed, that's the case. So you can see that while CD44, which is one of these markers, is almost universally positive in these cells, there are very small fractions in the MC38, about 2% on average, or 1 to 2%. And in the CT26, almost 4% of cells that were SCA1 positive. And to prove that these weren't even contaminating, we sorted these and we performed whole exome sequencing as well as RNA sequencing, and could validate that in the SCA1 positive cells, these were indeed MC38 cells. But what's interesting is the plasticity of these cells. And what we found is that if you if you try to purify these cells in MC38, what happened is we could never retain them, that over time, they would go back down to the 1 to 2% population. You could maybe purify them to about 50% by FACS sorting, but then if you just culture them, they drop right back down. Whereas in CT26, if you purify them, they're actually more persistent. And so we wondered, could there be some sort of cytokine factor that is responsible for maintaining them? And when we did cytokine profiling, as I mentioned, we saw IL-6 had shown up in the MDOTs. And what we did is we screened a whole panel of cytokines that were higher in CT26 and found that IL-6 was higher. And when we screened all these cytokines, indeed, it was actually IL-6. So that if you purify the MC38 cells without, and then try to culture them without IL-6, they drop back down. But in the presence of IL-6, you can statistically significantly maintain a higher proportion of these cells. And you can also see this even in the CT26 model, which had higher levels. Now you can really drive with IL-6 the proportion of these SCA1 positive cells. And in a variety of other lung models that have high SCA1, these are KRAS models that have high SCA1 to begin with, IL-6 clearly drives expansion of these cells. Consistent with the idea that IL-6, what was being induced, is actually allowing these cells to expand and persist. Conversely, I mentioned this TNF signature. And so we were wondered, because of that, what happens if you actually treat these cells with TNF, these SCA1 enriched cells? And what we actually saw was the opposite effect. And when you look at gene expression, you can see that TNF drives a lot of death genes in these cells, but does, as we picked up in our signature, have NF-kappa B survival genes, in particular two that I'm going to focus on, BERK2 and BERK3, that have actually come up previously in anti-PD-1, sensitizing to anti-PD-1 and CRISPR screens. And so when we just treat these for enriched cells, as I showed you, while IL-6 is able to expand these in both models, TNF actually kills them in both models. And what was interesting is that there's this tug of war in that in MC38, TNF wins out over IL-6, whereas in CT26, which is the more resistant type, IL-6 actually wins out over TNF in terms of allowing these cells to survive. And so we wondered, what is the potential therapeutic relevance of this, that while there's this tug of war, could you co-opt this TNF vulnerability? And so this comes to our collaboration with Novartis Oncology that they've been developing, as have a number of companies, these drugs, which are known as IAP antagonists, which interfere with BERK2 and 3 function. And a variety of other publications recently have shown that these can sensitize tumor cells to TNF. And what you can see here in the MC38 model, just on the top row here, is that TNF, again, kills these SCA1 positive cells, IL-6 expands them, and TNF wins out. But now in the presence of this BERK2/3 antagonist, basically you completely eradicate, in the presence of TNF, these SCA1 positive cells, so that even though there were still some cells here in the combination, they're completely destroyed. with the combination, and particularly in that CT26 model, where they were more resistant, there you saw the LCL161 could really help TNF win out over IL-6, but not completely eradicate the cells. And so the question is now, if you treat this MC38 model with this combination, what happens? And here I'm showing you the anti-PD-1 result that I showed you earlier in MC38, that you do see a shift in sensitivity, but rarely do you ever cure mice with PD-1 blockade in this MC38 model. And we think this could in part be to the subpopulations of tumor cells that persist. This just shows you that if you treat with this BRK2/3 antagonist by itself, LCL161, there's no effect. But what we found is that now you can start to achieve cures. Not perfect, but 7 out of 11 of mice are now cured with this combination. And again, in the interest of time, what we saw is that you saw the similar trend in the CT26 model with this combination therapy, arguing that if you can co-opt this TNF vulnerability of these persister cells, that you might be able to enhance cures. And so what to end this, and to go to the panel, in this part of the talk, the real point I want to make is that with current models that don't address the dynamic nature of checkpoint blockade, it's very hard to find these persister cells. And in fact, that's how they were discovered with EGFR therapy, was you needed cell line models, ex vivo models, to discover them. And we believe that these are relevant to human biology, and have now started to look in human tumors at these similar markers on checkpoint therapy. And the idea is that, similar to drug-tolerant persister cells, these immunotherapy-tolerant subclones can mediate anti-PD-1 resistance. And as I just mentioned, that because they're potentially more sensitive to TNF, even though they can be expanded by IL-6, that you could co-opt this vulnerability, in particular with these IEP antagonists. And it's there are actually parallels to the drug-tolerant persister cells, because in drug-tolerant persister cells, there's a lot of interest in targeting the BCL, the more canonical family members. But here what we found is that a TNF-related apoptotic vulnerability is uncovered. And so we think it's curious that in the immunotherapy context, that perhaps there's some shared ideas, but that it's really the apoptosis that governs TNF sensitivity that is more relevant. And so finally, I think I'm on the same page with my other panelists that incorporating function into precision immuno-oncology is a very important focus for the future. And I love that summary slide, and it sounds like we're going to also talk more about that every technology has its strengths and weaknesses, and that they would likely be very useful to address different questions. And I'm 100% on the same page that I didn't mean to criticize organoids and PDX models with their timescale. I actually do think they'll be useful and potentially practical in early stage patients, because one has the time to grow them and start to prepare if the patient relapses. And I do think, even though you lose the heterogeneity, that for this question of T cells, that if one can grow the T cells and find antigens, that could be very useful. But I also think that the technologies like the XPlant, the beautiful XPlant model that was described, and this organotypic spheroid models can continue to be refined, both in terms of readouts, how cells are cultured. We certainly have not answered all of the questions. But I think that ultimately, by truly personalizing cancer immunotherapy and really being able to test combination approaches like what's done in microbiology, that there will be a much more successful development into the clinic. And so finally, just to highlight people involved in this work, as I mentioned, Kartik Sehgal really led the project from my lab on the immunotherapy persister cells, and who's not on this slide because he now has his own lab, is Russ Jenkins, who developed the model in my lab as a postdoc, and is now at MGH with his own lab. And throughout all of this work, the Belfer Center has been instrumental. Cloud Powlitz, who's the director, Elena Ivanova, who has really helped in terms of most of the imaging methods, Patrick Lazat in terms of the immune profiling, and a number of others. There's a lot of bioinformatics support that is, as everyone appreciates, as required. This has been my funding. So thank you very much.
Okay, thanks, Dave, for the excellent and really thorough talk. And we can discuss a little bit about the model selection in the upcoming panel. And first of all, I have to apologize because I had my Williams mixed up. So Will Kaelin is the Nobelist from Dana-Farber, not Will Hahn, even though both are great scientists. And of course, I would give Nobelists to all the scientists if I could. And just a practical thing, we have around 300 participants in this seminar. So, and if you want to view the recording, it will be available in our website later. So in the interest of time, I'm just going to take a couple of the really good QA questions from the chat to Dave first, and then we can move on to the actual panel discussion. So I think really interesting question came from the audience. So what is actually the role of the microfluidics in the Barbie lab system? So in the other words, would you get the similar result if you would just have this kind of static ECM 3D culture gel thing?
Great question. So in terms of the microfluidics, it's true that for the current system, we haven't really employed the full potential in terms of introducing flow and vascularizing these models, which is what Roger Cam designed the system for. However, we did compare, if you take a standard 96 well plate and you use the same amount of collagen, same spheroids, all the same conditions and control, that we actually don't, you see a very small cytokine response in a static 96 well model, whereas in this system, we actually do see a much more robust response. And we think that in part, it does have to do with the way that the device is designed, that there's flow ports on both sides that allow free exchange of cytokines. But we actually haven't figured out the actual the mechanism as to why this is much better. But we've done the experiment and show that it actually does make a difference, this chip.
Okay, another kind of technical question. So you showed that you can also like treat the tumors with checkpoint inhibitors and interferon in the device. So how do you actually choose the drug concentrations that you use? Because I guess it's a bit tricky with this kind of drugs to choose.
Very good question. And so what we've picked is the Cmax of the PD-1 antibody, because again, in our case, we want to make sure that we're not underdosing in terms of seeing a response. So we don't go, well, anything that's super physiologic, but we basically pick when a patient receives pembrolizumab or nivolumab, what's the maximal concentration before it falls.
Right, that makes sense. Also, one more question about the tumor stroma. I'm a big fan of studying the stroma as well. So how well do you think that this kind of device or system addresses the fact that there's often this kind of dense stromal structure surrounding the tumors? Or could that be modeled somehow, for example, in this kind of system, in your opinion?
Great question. And all I can answer is that we know we have, we do capture fibroblasts. We haven't expanded them. So as opposed to other groups that sort of culture the tumor-associated fibroblasts, they're much higher numbers. So we're basically getting whatever is there, and they are a minority of the population. And again, we are reconstituting in collagen, type 1 collagen. We have played around with other matrices, fibrin mixtures, but right, I think one could certainly model this. And I think it's very interesting that the fact that we do see the matrix get dissolved. In fact, part of the reason we use fibrin is it can actually stabilize the matrix better than collagen. But understanding that biology, because there's so many different variables, we haven't tested that aspect, but it's a very good question.
Right. Okay, so maybe we can kind of move on officially to the panel. I see our panelists are popping up here. So we're here joined by the 3 speakers of the session, but also to get a bit more clinical perspective, in addition to Dave, of course, we're joined by Peeter Karihtala from Helsinki University Hospital, who's a research director in the Department of Oncology. And maybe Mirjami, if you could share that slide again. So we already saw from Jeroen's presentation this kind of, well, a discussion item. So this table that is kind of saying which models would be good or bad. And I really like how a lot of these talks addressed that there's no perfect model, right? So we just have to choose. And we all know that 2D cell lines have been extremely useful with their high throughput, low cost, and a good speed for like discovering pathways and even discovering drugs, but we still know that they're lacking on the extracellular matrix side and the 3D side. And we also know that the failure rate in the phase 2, especially the phase 2B clinical trials, is really bad. So where the efficacy is actually tested. So I guess my first question to all of the panelists is that, what do you think are the factors that you think is important when selecting a good model system for your research, and how do you define a good model system? And also, if you have any hot takes on good or bad model systems, the floor is yours. So I guess we can start with Dave, since he's already warmed up for the answering. So Dave, go ahead.
Yeah, I mean, I guess what I would say is where, thinking about what we focused on, which is the spheroid model, where are the limitations? And as very much what Jeroen mentioned is, especially with the patient material, is that is our bottleneck. And I think that's where, the throughput right at the top, the negative for explants, is that we're very limited by what we receive. And if you're not expanding it, you can only do a limited number of experiments. And so, but it really depends on the question. So again, here, if one of our questions is if we're, say, perturbed, we want to look at what I mentioned, what we're particularly interested in is interferon of sensitivity or sting agonism, that one can still test that and the priming in that context. Whereas we're also very interested now in the T cell biology, and if one wants to culture the T cells, it's very impractical to use our model. And so that's where actually the organoid or actually growing PDXs to have a bank of tissue to then be able to go back and screen your TILs. So I think for me, that's my very basic, from an IO perspective, that if I want to focus on finding antigens and T cells, that you need the organoid or the PDX model.
Okay, anybody feel free to chime in from the panel?
Well,
yeah.
for me, it's actually coming back to the reproducibility, because the reason why we are stuck with 2D cells and cell lines especially is because you can go back 20 years and you can repeat the same experiments that you did 20 years with drugs that were then available and do it today and say, is it better or worse? Okay, does it say anything about will the patient be cured? It's a different story. The translation up we know is bad. But the thing is that pharma is very persistent in their ways. So that's why we see all the models that are here plus organ on chips. Every pharma will use something else. And the reason is because very, very little is actually possible to reproduce. And so we are back to the reproducibility, which brings me back to the matrices where things like collagen and matrigel, every time you do it, you will be with a little different result. So I think you have to try to fix some of the things that you can. And the cells, when they are patient-derived, that's the one variable that you want to keep. You want to have variable patients or different sources of material to a certain standard of processing. But the rest you want to fix, so that when you have a question, you are actually not answering it with a mix of different answers. So which in part is due to the cells, part due to the matrix, part due to the variation in the growth factors, or way how you have handled it. And that's the points that I had in my presentation: the automation, the lean material, and then also the way you are handling it, so that then you can reproduce, you can validate. There are very few validated models out of I've been 20 years in this field, a lot of research, very few validated models. This is one of the reasons.
Yeah, actually, Johana, maybe to that there was an audience question, which maybe you can address, even though you're not working in pharma, but close to pharma. So do you think that pharma is actually moving their drug development to this kind of patient-derived models, or is it still too difficult?
I think the difficulty comes from patient consent. So it means that you have to have biobanks where you have cells with patient consent, and not only for one specific study, but something which would be a little bit wider. And then comes the question of legislation. So how easy or difficult it is to ship actual patient-derived. I think patient-derived cells are doing better than actual patient samples across borders or over the ocean. And that is something which I think we'll hopefully see positive developments. And meanwhile, again, I hope that this patient-derived would be a way forward. So you could have some panels of specific patient sample tissues. I mean, like lung QT, there has been a panel in Finland for some time. And that would be something that you could use to screen as a service at first, because if it's not possible to ship, you have to do it in-house. And then eventually you would have some transportation where you prepare the cells and ship them in room temperature, in incubated temperatures, to avoid, again, the freezing step, which brings a lot of uncertainties.
Yeah, okay, so maybe coming up in the future. Then I want to go more to the actual topic, so about the clinical translation. So maybe I will first address the question to Peter, and then David can kind of fill in if there's something. So what do you think is the reality of using this kind of patient-derived models in clinical diagnostics, maybe now and then maybe in the future? And also, like as a basic researcher, we often would like to know that what are the things in the wish list from clinicians to the basic lab researchers? So is there something that we can do to help to make it easier to make the clinical models better, or the translational models maybe? So Peter, if you want to start.
Yes, thank you. And thank you for great presentations. And it's clear that there are some obstacles, of course, before we can use these models in clinic. Well, the main issue is in the end, whether they have any survival benefit. That's the main question, which we have to resolve before they can be used in clinic, and we would need randomized study in the end. But David just greatly discussed about the static measurements problems with it, and when predicting response of IO compounds response, and also spatial heterogeneity is one important point in between different sites of metastasis, which should be kept in mind when doing these experiments. And tumors in different sites of body, they may be really different, also based on their genetic, but also based on their immunological composition. And that's one problem that we have to overcome somehow. Maybe, for example, using more early stage, smaller tumors with less heterogeneity is one approach. And for example, in breast cancer neoadjuvant or also in rectal cancer neoadjuvant settings can be used, where at least they can be a good start point to start with these models.
Yeah, that's an
excellent. But I see good future, but there is some work still to do.
Yeah, I don't think anybody so far addressed, like, okay, there's patient-derived tissue, but where is it actually coming from? Because it's relatively easy to get the primary tumors for your cultures, but then the metastases can be all over the body and be smaller and everything. So yeah, this is a really important point. Do you, Dave, have anything to add from the clinical perspective of using these in the future, maybe?
I would add that the heterogeneity is a great point that I didn't discuss, that in a stage 4 patient, the hope would be that, the advantages that checkpoint blockade in advanced stage does tend to respond, systemically, at least for a shared antigen, but the tumor microenvironment of what you're sampling is very critical. So I completely agree that in neoadjuvant setting, I think that the issue with actually translating, you can see that a lot of what we did was in mouse models, because we could control the experiment. We could actually validate in the mouse and ex vivo, whereas a lot of what's currently done with patient samples and the reproducibility, et cetera, as Johana is sort of getting samples, and you can't control whether the patient is actually being treated with that therapy. I think the only way to really move forward would be to incorporate this into actual early phase clinical trials. meaning that on the trial, you mandate a biopsy to keep it alive and then test ex vivo and then build that into a phase 1 type design and see if, can you correlate? And there are these adaptive trials that a number of companies are doing, for example, in the stage 4 setting where they're testing 3 different IO combinations, again, with static biomarkers. But if one could realistically take the biopsy, test those 3 combinations ex vivo, and then randomize the patient to the effective one ex vivo, then you could actually study it and see, does it work? But that's what you're required to prove.
Yeah, that's definitely something we're missing right now, because everybody's just hyping the organoids and these models, but then we don't really have the data yet if it's really as predictive as we think. Maybe from Jeroen, I was actually just coming.
I think one important thing is that what David already mentioned in his talk is the time aspect. So I've heard David Barbie from the Cold Spring Harbor talk many times, and he is developing these organoids for pancreatic cancer, and he can predict, according to him, quite well. The problem is it takes him 3 to 4 weeks to get the results, and according to him, this is not sustainable in the clinic. Maybe for a second line treatment, but the doctor simply does not want to wait a month for the results of the organoid cultures to tell him or her what to treat with. So it needs to be faster. I think that this is an important drawback. And as David mentioned, if these are on these primary tumors or that have been operated upon, but with pancreatic cancer, this is apparently not the case. So treatment needs to be almost immediate.
Yeah, actually, we're running a little bit late on the time, but we can maybe continue for a couple of more minutes if there's time. There was actually an audience question for Jeroen. So the audience wanted to know that how about these explants that you introduced, that how long can they actually be kept in the lab, and how long is the microenvironment sustained? And would you say that this is a good IO research tool? We saw some of the immune cells in the cultures, but what's kind of your take home from the basic research side here?
Well, so I think the data that we've shown and some unpublished or preliminary data that we have show that it's a good tool. Obviously, how well it reflects what happens in the patients, for that we will need to do what David was saying, real trials. We keep them generally, we can grow these explants for a really long time, we can keep them alive, but we do know that this affects, we don't really see hypoxia happening, but we do see some changes in the cancer. They change just their subtype, or go from basal to luminal, that kind of thing, but not within a week. And it's anyway, for practical reasons, it's just better to keep it within a week. Also, we know that for a week, as David mentioned, we keep the immune system as it is. We've not tested this for longer periods, so we don't know if then changes start to happen. Probably they will.
Right.
So I heard that Peter has raised his hand, so maybe.
Okay. Sorry, I didn't see. So Peter, go ahead.
Yeah, just a minor comment about the gap between biopsy and result from it. And well, if we are speaking about metastatic disease, of course, 4 weeks is too long time to wait. But in adjuvant setting, for example, there is from surgery to the beginning of adjuvant treatment, there is usually 4 even more weeks. And in adjuvant setting, this could be more feasible. Of course, drug trials are usually begun with metastatic setting. And also, for example, in neoadjuvant breast cancer setting, usually there is any primary drug resistance in most breast cancers in neoadjuvant setting. And if we get that predictive result after 4 to 6 weeks after beginning of the treatment, we still have time to change the treatment before the secondary resistance appears. That is just my minor comment.
Maybe I can add on that really briefly, is that the problem when I completely agree that adjuvant, neoadjuvant is the setting in which one can minimize heterogeneity. But as the readout is, the other reason stage 4 is easier as a readout is you can see shrinkage of a tumor, but because you're already, trying to cure the patient with surgery, the readout is much more delayed, it's relapse. But if one could developing circulating tumor DNA markers for MRD, which are now coming online. One could use those as your readout.
Okay, maybe to wrap up the session, I will
maybe
ask.
Johana raised her hand, so maybe we give the floor one more time.
Okay, yeah, I'm just worried about the timing, but if the panelists still have time, we can continue for a couple more things.
Just one this kind of commercial aspect again. I'm sorry, I'm on that side, but the problem with these, whether it's treatment or diagnostic of a single patient, the problem is always who pays. And this is not something which is now kind of like localized trials, or it's not something that would be even nationwide or European or American, and that's going to always be a problem if there is no reimbursement, a clear reimbursement path, how to make it more stratified as a plan. This will remain as a marginal thing to bring this into real treatments. This requires also some work on that part.
Yeah, and it's kind of worrying that it could be something that you get in a private hospital if you're really rich, but then if you're not, then you're not going to get that kind of fancy diagnostic tools. But again, maybe to wrap up, I will ask one more common question. So I guess we can all agree, based on what all of you have said, that we have to make the in vitro research models more and more complex. But I also see there's a lot of problemacy there, because then it also means that our lab work is getting more and more complex, and we need to be able to do drug screening and all these things in these systems. So what do you think would be the solution? And do you see that this kind of systems can ever be made in kind of high throughput research models in the future?
So
Well,
go ahead, Johana.
I can get started. So in my opinion, this is again a question. We have already seen nice results where you have patient samples stored for processing in our material, so that you can automate it a bit. So instead of when you have a patient coming from surgery, immediately starting working with the sample, which rarely happens, you have an intermediate step where you kind of store the biopsies, whatever is the tumor, and then process them all on one day, same technique, same way, and then the process further on is possible to be automated. Otherwise, it's always going to be kind of very, very manual. And then I think the second part is that when you have then this kind of, let's say, more automated approaches, it is also more likely to become more mainstream, because then you are taking out the uncertainties from the process. So that's my take on it.
Jeroen, do you want to continue?
I think that there's a clear, I think, explants are not suited. If you have a drug or a small drug panel, explants are suited for testing, like in David's example. But organoids are suited for earlier work, when you're still trying to screen a larger number of compounds, because you cannot really realistically do that with explants, at least not with the patient-derived ones. So I feel that it's a matter of selecting the right tools at the right stage. So you start with something simple, so either organoids or then co-culture of cell lines with T cells, where you screen for drugs, but you don't select one but a panel, and you narrow that panel down as you go more complex. So that's how I see the drug development being, and definitely mice have still a role here. That you then go with a few of the selected drugs also into mice, and ideally the drugs work on both explants, which are then the human component, but also on the mice, because obviously the mice have a whole circulation and lymph nodes and all of those things that are obviously very important.
Yeah, I kind of really hope to see in the future that we could create this kind of human on chip type of model, so we could at least reduce the animal experiments that we have to do, but I guess the time is not right for that yet. I think because we're running out of time, I really want to thank about all the great audience questions. Unfortunately, we couldn't address all of them, but at least some of them today. And I want to thank for all the great talks, and I want to thank the panelists also. And this recording will be again in our website and LinkedIn. And this is a bi-monthly seminar series, so we're going to continue in 2 months with some other interesting topic. And yeah, thanks for everybody and see you later.